Good morning, everyone, and welcome back. We gather again to thank the Lord for all His blessings. In the name of the Father, the Son, and the Holy Spirit. Amen. Blessed Trinity, we love you, we adore you, and we thank you for your many blessings. We ask that you continue to be with us as we delve into the issues of life, human life, and the life that surrounds us. May we be able to develop a profound understanding of our nature and so be able to uphold the dignity and the sanctity of human life, and also the dignity of the nature that surrounds us, so that we may protect it as good stewards. And we ask this in the name of Christ our Lord. Amen. Father, Son, and Holy Spirit. Very well. So welcome back. This is the course on fundamental bioethics, and today's topic is on origins. Okay. Before we get into the actual topic, any questions from previous lectures or comments? Anything at all? No? Okay. Let's try to adjust the lights here a little bit so that we have more visibility on the screen. Good morning. Welcome yourself. All right. Is that going to be too dark or no? Fine. I think, yeah, it's okay. Okay, if you want to move in a little bit or over there, or space over there. You can also use this one if you want. Did you get the handouts? No, she didn't. This one. There's two. You want to have an extra? Everybody get two? No, I got one. Oh, okay. So you missed. Okay. So here are those. This is that one? Yeah. That one. Yeah. Oh. So this one and this one. Sorry about the smear on the print. That has to do with the photocopy there. I need to change the setting or the toner. Okay, so this little video is going to be low or light, but that's all right. This can be minimized. And this can be minimized. Let me pull up then the handouts that I gave you. We'll start with this one. So far, we have been looking at the evidence for evolution. Let me back up one more and just to review that this course is to develop a theological anthropology, right? In other words, a vision of the human person that is as deep, as ample as possible, being respectful both of the scientific evidence of the nature that surrounds us, so pretty much the material world that we live in and as living creatures, and at the same time our faith, which takes us beyond the physical into essentially the metaphysical, that is not measurable in the sense that the physical realities that surround us, including our own life, is measurable, right? And so we want to do something like it has been said for years in the church, reason illumined by faith. Reason illumined by faith goes all the way back to St. Augustine, and both Pope John Paul II and Pope Benedict wrote a lot about the interaction and the dynamic dialogue between reason and faith, or science and religion and so forth. Even John Paul II wrote a whole encyclical, Fides et Ratio, Faith and Reason, which is very beautiful encyclical. Okay, so the first part of the course then is devoted more to the scientific evidence of life, and particularly human life, and how we come about. And then the second half will be incorporating or integrating, if you will, our faith belief from sacred scripture and tradition, as we have those two foundations of our faith, right? The sacred scriptures and the tradition of the church of the 2,000 years that we have. So looking then at the process that is a process that is occurring in nature all the time, even though it's long-term, is this process of evolution, okay? Evolution, which is the six aspects that we have from evolution that are evidence for evolution, beginning with the fossil record, which we cannot deny. These are organisms that existed in the past sometime, typically millions of years ago. So we know that the Earth is about 4.5 billion years old, 4.5 billion years old. That's 10 to the 9th, right? And that some of these fossils are very ancient indeed, and they match up with the geological strata of the Earth. And we can then put a date on the fossil, or the fossil record. And then there are other lines of evidence, if you will, that point toward this process of evolution. Of course, as we stand today and look around and look out the window into nature, at this point in our time, these are the products of evolution, including ourselves, but it's an ongoing process. But because it's so long-term, right, then it's hard to observe, let's say experimentally in a lab or something like that, and say, okay, this 15 minutes, this next 15 minutes, there's evolution happening, because it's typically measured in millions of years, and none of us live that long. So we have to pretty much go by the accumulated evidence that we see around us. So again, using our reasoning to infer this process. And so the lines of evidence, the morphological similarities of derived characteristics, also the comparative embryology of not only vertebrates, but also other embryos as the different species develop into maturity, vestigial organs, the biogeography, right, that we see the diversity, but at the same time relatedness of animals and plants that live in totally isolated geographical regions, like continents are separated by entire oceans. And finally, the more recent one, if you will, in the past few decades, but the more comprehensive one is the whole issue of molecular evidence for evolution, because then we can go into homologies, percentage homology, and that you can see that is very quantitative and qualitative at the same time, right? So qualitative meaning that we could look at regions of DNA or genes, or their expression, which is going to be some kind of protein, functional protein or structural protein. That's on the qualitative side, what type of gene it is, what type of protein. And quantitative, but then we can measure the percentage homology down to the base pair level, all right? And by doing all that, we come up with, the scientists have come up with these phylogenetic trees, and also in the classification of the species, the living species, of which I've mentioned scientists have classified approximately 2 million. Of how many species, did I mention how many species are estimated to live on Earth today?
2 million?
Well, these 2 million actually exist and have been classified, right? But is that 100%? You think that we have discovered all the animals, plants, fungi, and especially bacteria and archaea that live on Earth? No, our intuition tells us, because there are discoveries all the time, okay, our intuition tells us that we have discovered mostly the large plants and animals that we can see and so forth, and some of the bacteria and archaea that we have been able to identify. But it's estimated that this 2 million is only about 5 to 10% of the living species today. So we're talking anywhere between 10 to 20 million species estimated to be living today. Now, again, what is your intuition about that other 90 to 95% of species that we haven't discovered yet? Do you think that they're large and kind of stick around and with many individuals in the populations all over the place? No. They're very secretive. They're hiding. Every now and then, every few years, you get a report of a new animal that is found in a jungle somewhere, very recluse, right? But that's very rare. It's estimated that the vast majority of these species that we haven't discovered yet are, first of all, microscopic, and secondly, they live in the deep ocean, like the ocean floor. Because again, consider the ocean floor, everything that's dropping down, that is raining down on the ocean floor, literally, sediments for millions and millions of years. Imagine a whale that eventually dies, right? After 60, 80, 100 years, I don't know how long they live, but eventually all whales die. And that carcass is going to go to the bottom of the ocean. And there it's going to rot and decay and also be picked away by all kinds of crustaceans and other creatures that would bite at it. And decay and be eaten up by algae, by bacteria. Eventually a skeleton will be left over, some more sediment will fall on top of it, and maybe in a few more millions of years they make a fossil. So imagine all the fossils potentially that are on the ocean floor. But that's 1 or 2 miles deep. You know, the pressure is an exorbitant pressure, and it's very difficult to get there with machinery, let alone an actual live human being. It's mostly these little submarines that are being sent down there, okay, with cameras and lights and so forth. And all kinds of life has been detected. But at the macroscopic level, it must be immense. One can intuit, because you have a medium that is more or less isotonic, meaning that the salt water is more or less equivalent to the density of cells, right? And so there's a dynamic interaction there, and it's just full of nutrients also, no light, of course, so they would have to be consumers, or they go into a process called chemosynthesis. I'll talk more about that later today in the lecture, but independent from a light source, a life chain that is independent from the light source, from natural light, which is sunlight. Anyway, all this to say that the vast majority of these creatures that have not been discovered yet are likely at the ocean bottom or other parts of the world too, okay? In what is known as microclimates. For example, very tiny sectors of a pond or even a lake, typically in a water medium, in a liquid medium. So back to the molecular evidence for evolution allows us to make these phylogenetic trees that are, as you can see, very comprehensive with lots of branching. But three main domains that we have talked about: the archaea, the bacteria, and the eukarya. And today I'm going to give you a little more substance of how to distinguish these. We'll have to focus the distinction between the bacteria and the archaea is going to be on the membrane, because they don't have a true nucleus. They don't have a fully organized nucleus, especially with a membrane, right? And the issue of the eukaryotes is going to be precisely the presence of a nucleus that is enclosed in a membrane. So it's at the very basic level of structure, if you will, of structure of the cell that sets these three domains apart. And they're mutually exclusive, all right? So that these 10 to 20 million creatures, or at least the 2 million, let's stay with what we have discovered so far. The 2 million are either eukarya, archaea, or bacteria. However, when we go back, right, you see that they had at least the discretion to leave these trunks of the branches outside of the set, so that they may have been some common ancestry here, some common characteristics that may have had at least the passage from bacteria to archaea, because this is implying a single origin. It's implying a single origin of a single individual species, the first common ancestor, right? The first, not the nearest common ancestor, because the nearest, excuse me, the nearest common ancestor would be the last branching. Okay, for example, let's look at animals here. The least common ancestor, the nearest common ancestor between animals and quanoflagellates. What are quanoflagellates? Anyone? First of all, what's your intuition? Are they large or small? Yeah, they're microscopic, all right? And they have this flagellum, because that's our common ancestor with all animals. Quanoflagellates. Let's look at an image here. Right, they're microscopic and they have these flagella that wiggle, and many times they form colonies, small colonies, colonial, okay? And it's a single cell. But when they form a colony, then it's an adaptation of a single cell, because eventually those cells could specialize and begin to form multicellular tissue. Anyway, my point with this here was simply that, where was it? Here? Yeah. That's our nearest common ancestor between these quanoflagellates that exist today, right? These little guys, and all of the animals that exist today, beginning with the sponges as the most primitive ones, okay, or ancient, all the way through all of the invertebrates to mammals, and culminating with the human as the most recent mammal, basically, and the most advanced mammal also. Okay. So this, again, is it can be put together by this molecular evidence of percentage homology. Let's go back now here. Today, we're talking about 3 origins then, but very briefly, the first and the last origin, I'm just going to mention it briefly, which is the origin of the universe and the origin of humans. Okay. So that one and this one. As we can say, really, as far as we're concerned, for meaning and importance to us as the human species, the first origin and the most recent origin, if you will. First origin of the universe, the whole shamil, all right? Everything that is material, including energy, including the dark matter and the dark energy that is estimated out there, up until the most recent significant origin for us, which is the origin of the Homo genus at the level of genus, because we have fossil records of several different species of humans. And the big question is there, for example, did they have a soul or not? You know, did they have the possibility of eternal life, which would make them much closer to us? And if not, then it would make them much more distant to us, okay, precursors, but not fully human in our human context of today of eternal life. So this one I'll talk in the second half of the course. And then this origin of universe we'll see in more detail in the environmental bioethics course in the summertime, okay? So we're gonna focus mostly today on the origin of life, all of life, okay? for the rest of the time. So if there are no questions, I'm going to move forward. What do we have? Briefly, that the origin of the universe, the current model, the most respected model, if you will, for scientists is this Big Bang in common terms, in popular terms, which mathematically is called the definition of a point, right? Unidimensional point, and in physics they call it singularity. And popularly, this massive explosion of all matter and energy that forms the universe about 14 billion years ago, approximately 14 billion years ago. And I'll end with this on this part, is that's an estimation, that's an approximation that is based on what is known as mostly the background radiation that astrophysicists, and actually this area is more cosmology, right? In cosmology, they have been able to detect perhaps some remnants of that original Big Bang, and that's called the background radiation. And the estimated age of that background radiation is approximately 14 billion years, all right? 13.8 or something like that. So give or take a few million years back and forth. I'm not going to say anything more about that. As you can see there from the diagram, the caricature, there's been progress and dynamic development and progress, which I'm not going to get into. But then the Earth itself is very recent compared to those 14 billion years. The planetary systems and the solar systems and the galaxies, which would be at the edge here, excuse me, it's estimated to have happened about 4 billion years ago, or 4.5 billion years, 4.5 billion years, by a process called accretion, which is accumulation of the debris from the explosion, material debris, that started to coalesce, started to come together, mostly by gravitational force and collision, and forming this larger and larger ball. As the ball gets larger, there's a synergy here that as the ball gets larger, it is exerting a greater gravitational field and pull, whereas not only is it attracting more meteorites and asteroids that are flying around, just debris basically, but also internally is so hot that it's white hot, beyond red hot, and this material is molten, becomes liquid, and as it's liquid, then it's down to the level of the elements in the periodic table. They are actually coalescing, and they are differentiating by density, so that the more dense will go right to the core and form a dense core of the most abundant material, which is iron, and which becomes so dense that it eventually solidifies. But I'm going to leave it at that now, just to know that the process is called accretion, and it's a fundamental process of planetary formation, from the formation of the planets from solar systems. And this has been kind of observed, or at least deduced, from other planetary systems, also other solar systems that have been discovered over the years. Okay. So 4 to 4.5 billion years ago, the formation of the Earth itself and the planets of our solar system. Okay, and the human evolution, just from the sake of completion, of at the level of the genus Homo. All right, about 2 million years ago, maybe, and just to show some of the fossil record that has been found there. Okay. Now I'm going to zero back in to the beginning of life as such. So here, this section. And the reverse of this sheet. So this is kind of very general outline for today's lecture. And we'll cover this origin and development of life on Earth. In the first half of the course, the first half of the lecture. In the second half, we'll look at a hypothetical situation of how life would actually originate from inorganic material, okay? From inorganic material, in other words, inorganic elements and compounds, to organic materials, the 4 types of macromolecules: carbohydrates, lipids, proteins, and nucleic acids, DNA and RNA. And then that's the first jump, and the second jump would be from that organic material to a protocell, to the beginning of an actual organized metabolism, all right, going on, which would be hypothetically the first protocell, the oldest common ancestor of all life. So that's for the second half of the lecture. So don't look at the backside of this yet, basically. Now I'm going to be following, I don't need this one anymore. Well, I'll leave it on the background. I'm going to be following Meyer's chapter 3. Okay. So if you have the book or you took notes or made summaries, I also gave you my summary of Meyer there. So basically your task is going to be to try to integrate Meyer's summary with the lecture. But I'm pretty much going to follow this diagram so you can, this scheme, and you can see it there on the handout, right? And I'll send you this electronically also so you can work with it. Basically, the front part of this Meyer chapter 3 summarizes the chapter in 1 page, and then the backside is the geologic eras, or eons first, and then eras, where it points out this sequence of mass extinctions and radiations that follow in a sequence. All right. Okay, so bottom line, let's zero in on Meyer chapter 3. We see that accretion, he's putting it at 4.6 billion years ago, give or take 500 million years back and forth, 500 million years back and forth. All right. Fairly recently after that, fairly soon after that, there is already some evidence, fossil evidence of life on Earth, which is amazing. 3.8 and even into 4.1 billion years of fossil records. It is amazing. Take a look at this. Here we have what is known as zircon crystals. All right, zircon crystals have some uranium in them, and it's the high isotope of uranium, which is uranium-238. The other uranium that you saw for half-life was uranium-235, I believe. It's an isotope, and that one has, remember, the half-life of about 700 million years, right? 700, which is not even 1 billion. But this more reactive, more radioactive uranium, all right, 238, has a 4.5 billion year half-life. Exactly. So basically, what is that telling you? 4.5 billion years, and the Earth is about 4 or 5 million years ago, billion years. So basically, this was one of the first elements that formed back then, these isotopes, okay, at the very beginning of the formation of the Earth. Anyway, these zircon crystals then have some of this uranium, and that's how they have been dated, back to anywhere between 3.8 to 4.4 billion years ago. But the interesting thing is that they have carbon. And carbon, they have graphite, a type of carbon, right, that is used, for example, in the lead in pencils. All right, that's graphite. That is estimated to come from organic material. In other words, this is organic carbon, if you will. This may have been produced by some living organism. Okay, because carbon is one of the 2 necessary molecules or elements for organic material. All living things, let me back up a little bit, I'm a little dispersed today, but all living organisms or living tissue is made up of organic matter, right? In contrast to inorganic matter. So that's one big division for life, if you will. Living tissue, living organs and systems and individuals were made up of organic matter. And there are two elements that have to be present for organic matter, all right? And that is carbon, and what's the other one? Hydrogen. Okay, so carbon and hydrogen. So for example, water, what is the molecular formula of water?
H2O.
H2O. It's got hydrogen, right? Does it have any carbon?
No.
No carbon. So water is not an organic compound, it's not an organic molecule. And yet, water is necessary for life. We are anywhere between 70 to 90% water, depending if we're very old or very young, right? Because we lose water over time, over age. But water is necessary for life, but water itself is not an organic compound, okay? Well, how about CO2? CO2 is actually a product of respiration, right? We exhale CO2. So carbon dioxide, CO2, give it away. All right, so carbon dioxide has carbon, right? But does it have hydrogen? No hydrogen. So carbon dioxide is a product of metabolism, of cellular respiration, but it's not an organic compound. It's inorganic. Mm-hmm. And yet, when we combine, not we, but producers, combine water with carbon dioxide, they make another molecule that is organic. What is that molecule? Well, that's the precursor, yes. But I'm thinking producers, in other words, photosynthesis, animals that photosynthesize, the process of photosynthesis is the opposite of cellular respiration, right? Mm-hmm. They take water typically from the roots, from the soil, and they take carbon dioxide from the atmosphere, the carbon dioxide that is in the atmosphere that we're breathing right now, combine those two molecules to make an organic molecule. Well, oxygen is a byproduct, but the real thing for photosynthesis is glucose, carbohydrate, that organic molecule. C6H12O6. You see, so all the bonds are saturated with the hydrogen. That's what hydrogen does basically. An organic molecule saturates the bonds, or if it's unsaturated, then it's a double bond. It can take more saturation of hydrogen. Basically, glucose, C6H12O6, right? Glucose is an organic molecule, and it's made up of two inorganic molecules by a very living process of photosynthesis, which is really what allows for life on Earth today. And that's why photosynthetic individuals are called producers, because they produce food, they produce glucose, right? We, on the other hand, are consumers, so we are consumers of the food that the producers produce. At any rate, this issue of carbon and graphite is estimated to come from living organisms, and it can be found in zircon crystals that can be dated back to about 4.5 billion years ago. So it's estimated that life began anywhere around that time, 4 billion years ago. But at the same time, the Earth had to be cool enough to sustain life. But again, I want you to be thinking functionally, because, you know, this issue of function and structure, another little aside, I diverge like this back and forth, but consider this. What do we see? What do we see? We see, so most of our information comes through our eyes, right? And we see mostly structures, things, whether living things or non-living things, but we see structures. But it's important to train our minds to try to discern the function of what we're seeing, and not just stay with the structure, okay? So functionally gives us an insight into what's going on around us, and including ourselves, our own body. Organically, that function we call metabolism, all right? So when we see these structures, we have to intuit behind the scenes that there was some functional organisms to have made this graphite. Oh, yes. So functionally, at the crust of the Earth. So remember accretion, and it was a ball of fire essentially. At first it was white hot, and then it started cooling down to red hot, but it's still molten lava. And what can live on lava? Imagine the lava that is coming out of the volcanoes where it happened there in Hawaii over the summer, right? That it was engulfing houses and cars, and everything was being melted down into becoming part of the lava. Do you think that any life can live in lava? No, but why? But the heat organically or biologically, you know what that heat does? It vaporizes water. And so we cannot have, that's why water vapor is used to sterilize equipment, right? High pressure, the autoclave has water vapor, high pressure, and high temperature, because we need liquid water for metabolism. We need liquid water, and liquid water only exists within a range of temperature, which is what range? In centigrades, that's what establishes the centigrade scale, all right, or the Celsius scale, from 0 centigrade to 100 centigrade. That's the scale, and it's a metric scale, as you can see, all right? So that below 0, under standard pressure, okay, under ambient pressure or 1 atmosphere of pressure, in other words, more or less sea level, at 0 degrees centigrade, what happens? That is an interface for the state of water, pure water, right? From liquid to solid, right? To liquid to ice or solid. And then at the other end of that scale, at 100, that same water goes from liquid to vapor to gas, right? So the 3 states, solid, liquid, and gas. And that the window then, if metabolism is essentially a dynamic soup of chemicals that is interacting inside our bodies, inside our cells, and everything that is living, then that water better be liquid. But it cannot be too cold, because the metabolism then is going to slow down. It'll be too slow. And in fact, that happens to animals up north. They go into hibernation. They stay alive or dormant, right? They stay alive, but the metabolism is dormant, is almost shut down, not totally shut down. At the opposite end, if it gets too hot, then we're going to burn up. In other words, literally too much metabolism. Okay, and also at some point, the structures, mostly of the proteins, are going to denature. And so they're going to lose the active site of the protein and all that. They will become non-functional. So the window between 100 and 0 is even narrower for metabolism in centigrades, all right? And it's around what? From the 20s to the 30s is more or less the window in centigrades, right? 20s to 30s degrees centigrade is more or less the window for life, for metabolism to be more or less comfortable. To the point that we have an internal metabolism at what temperature? 37 centigrade. Now we can translate that into Fahrenheit, which is not a metric scale, and it's an awkward scale because it's not based on water, but it's based, I think, on mercury somehow. And what is this? What is the? So we can make a translation into Fahrenheit with our body temperature, which is the thermometer. You who are moms, and on the children before the temperature, okay, before they get a temperature, it is what? 98.6, if you want to, you know, but around there. But, you know, a temperature of 100 is Fahrenheit is already warm. 104 degrees is extremely hot. And that's not even 10 degrees higher than normal body temperature. On the other hand, here now, there may be what? It feels to me like
69.
Yeah, it feels below 70, right? It feels below 70. Yes, it does use the apparatus. They don't work, but anyway. It's the hottest, and press that button, and we'll see what happens. So look at that, just 20, less than 20 degrees below, you know, 15 degrees already is uncomfortable. So it's really a narrow range for life. It's a narrow range of temperature. It's a narrow range, very narrow range of conditions. And this planet that we call Earth happens to be at the right distance from the sun to have liquid water and has cooled down enough internally, to have even that narrower range of life, okay? And so that's why the oceans are so important, because they are stabilized, they stabilize the temperature on Earth. Kind of like a temperature buffer. All right. Anyway, let's go to Australia for a moment. And on the coast, there are these strange rocks, which are actually fossilized bacteria. Okay. And they're called stromatolites. And they are layers. You see these layers. But these layers are not just sediment layers. It turns out that when this is observed under the microscope, these sediment layers are actually estimated to be primitive bacteria or archaea that got trapped in the sedimentation. Okay, so it's not just sediment. This was living organic material at one point, and these rocks have been dated to around 4 billion years ago. So very early on, we see that life already emerges, but we need, of course, a crust of the Earth that is cool enough, that the Earth is, first of all, solid. Secondly, that there is this liquid water that has accumulated, mostly the ocean basins, which is 3/4 of the Earth's surface. Okay. So really, I'm starting not with the origin of life, but with early life. You see the distinction? We're going to do the hypothetical origin of life in the second part of the lecture. But here, I'm starting with the evidence for life already, All right, so these sediments are estimated to be of archaea or bacteria, which are known as prokaryotes. And prokaryotes means karyo is a reference to nucleus in Greek, right? Remember, you are also learning a little bit of Greek and Latin along the way. Karyo is a reference to nucleus in Greek, and pro is not yet. In other words, not an organized. It's not an organized, a precursor. That's what I was looking for. Pro, you can interpret as precursor. Okay. There's another analogy, for example, the pronuclei of the egg and the sperm. All right. They are pronuclei because they're haploid. And when they fuse in fertilization, the egg and the sperm, then those two pronuclei fuse also together to make a diploid nucleus. All right, so the pro is a reference to a precursor. So these are precursors to nuclei. Basically, there's a concentration of DNA or RNA, depending what bacteria or archaea is. A few actually have RNA, but the vast majority have DNA as the inheritance material, as we saw from last lecture, and it's concentrated in a region of the bacterium or the archaea inside, but it doesn't have a membrane, it doesn't have a nuclear membrane, which, by the way, is a double membrane, which is very interesting. Just like the cell membrane itself is a double membrane. So there's no, we say that the nucleus is not organized, and that's why it's a pronucleus. All right, it's just an area of concentration of the DNA, whether it's bacteria or archaea, but there's no actual cellular membrane to protect that nucleus, for example, from radiation. That's why bacteria and archaea are more susceptible to radiation and chemical attack, if you will, because the DNA is more vulnerable. It's not protected by a second layer of membrane, which would be the nuclear layer, the nuclear membrane, in addition to the cell membrane itself. All right, but in that sense, both archaea and bacteria are similar in that neither one has a nuclear membrane, and therefore they are classified as prokaryotes. And yet, they have a very big distinction between the two, all right? And that's why it's estimated that archaea are more primitive than bacteria. Because they lack, this is kind of a magnification here diagram of the cell membrane, the actual cell membrane. Let me see if I can, oops. Okay, down here, this is the outer membrane with all the receptors diagram there, and this would be the inner membrane, okay? And there's a layer in between that is called Peptidoglycan Layer, which has proteins and lipids. Bacteria have that layer, but archaea don't have that layer of peptidoglycans. They're missing that layer. And so that's why they're considered to be more primitive, in the sense that this layer was probably developed later. Or there's another school that says, no, it was the other way around. The bacteria were first, and then these archaea lost that layer. And so they would place the bacteria before. For example, I was noticing that on this diagram, they put the root inside the bacteria, not the archaea. But this is totally speculative where this root is, whether it's in the bacteria or the archaea, or actually outside to a primitive organism that was neither, right? It was neither a bacterium or an archaeum. All right, so that's the basic distinction between these two, and I'm pointing it out because this one layer makes the distinction, the presence or absence of this intermediate layer on the cell membrane, makes a distinction between two domains, two of the three domains. So imagine how basic it is, right? It makes the distinction between two of the three domains of all life, the highest category possible, the highest grouping, the more diverse, if you will, more inclusive. Okay, my point? So it's a very significant layer, to have it or not. At any rate, we move forward then. Just pointing out that bacteria and archaea, many of them are also extremophiles. Now we go to Latin. Philia, philos, is a liking. For example, friendship comes from there, all right? Philos is a liking, and they like extreme conditions. These bacteria and archaea, they like extreme conditions. For example, excuse me, some of them like extreme salt, as in the Dead Sea, right? The Dead Sea, which is in Jerusalem. And this one, actually, you can barely see this tube pumping out. This is probably from nitrates and phosphates, all right? But it's an extreme concentration of minerals. And some of these love to live in those areas of extreme salt or extreme minerals. They're called halophiles, because halo or halo, depending if you're American or British, halophiles or halophiles is a reference to salt. All right? In fact, somehow the halo has to do with salt crystals reflecting the light or something like that. Others like it hot, very hot, close to boiling, all right? And so those are thermophiles. And they live typically around geysers or geysers. How do you pronounce geyser or geyser? Geyser, okay, around geysers, where this is very hot steam coming out that is liquefying when it cools down in the atmosphere. And many times they are both thermophiles and halophiles because they have a lot of sulfur or other contents, other mineral contents, okay? Some are like it totally dry. They can live in practically close to zero humidity. Xerophiles, zero is a reference to the desert, to dryness. Turn this out. Yeah. okay? And others actually are radio resistant. They can live under UV, for example, and tolerate high amounts of radioactivity. Now, what's interesting about these is that some of them actually, a few species have been found, or types of bacteria have been found that gather several of these characteristics, okay? And not too many have been found, I think maybe one or two, but those are called Extremophiles. And those are very nasty because basically we haven't figured out a way to kill them yet. And that's the possibility of the terraforming that I was talking about. These extremophiles would possibly tolerate the harsh conditions that would exist in another planet with very little or no atmosphere. Okay. Anyway, speculation, but the polyextremophiles have been found. Let's try that briefly. Oh, the tardigrade? No. There was... yeah, okay, well, there would be around the sea vents that I'm going to be talking about soon. Yeah, I think the tardigrade can actually live with little or no oxygen, anaerobic conditions. All right, so what is the point of all this? That these prokaryotes, especially the extremophiles, could have been the precursors of the contemporary bacteria and archaea. In other words, they could have lived in the harsh conditions of the primitive Earth, which did not have oxygen, was a reducing atmosphere. I'll get to that in a minute. But basically it was toxic for us today. We would consider that very harsh conditions, and little cloud cover and such. Maybe there was much more UV exposure. It was also hotter and more minerals and so forth. So these bacteria could have been precursors of these bacteria, could have gotten the life chain started. Then there are the cyanobacteria. Okay, cyanobacteria. I can tell you, when I was studying biology in the 70s, the cyanobacteria were still classified as algae. They were classified as algae because they were called blue-green algae, I remember, because they can photosynthesize. However, aside the fact that they can photosynthesize, they did not have the other characteristics of true algae, especially by way of the cell membrane, and they did not have a true nucleus, which algae have a true nucleus. So algae are eukaryotes. And therefore, they were demoted, if you will, to bacteria. But the cyano is a reference to a blue-green color, which is the photosynthetic pigments. Now, what's interesting about these photosynthetic bacteria is that there seems to have been a huge bloom of these bacteria early on, around 3.8 billion years ago, when the atmosphere was originally not that conducive. The primitive atmosphere was known as a reducing atmosphere. Okay? A reducing atmosphere is that there is no oxygen. So in the redox reaction, remember redox reaction, let me see here. It's an equilibrium between reduction and oxidation, back and forth. See if they just show the formula. Right. Okay, so here we go. For example, using iron plus oxygen, it rusts, right? Iron rusts. Okay, rust scientifically is known as oxidation, and you get iron oxide. And this is a reduced compound. So if we break up the iron oxide, we end up with, okay, this is not balanced. We see it's not balanced. It has to be balanced. But the point is that adding the oxygen to the iron reduces. Taking out, adding the oxygen to the iron, it oxidizes the iron. Taking out the oxygen, separating them, is reducing, going in the opposite direction, is reducing the two elements. The issue with the reduced atmosphere, primitive atmosphere of Earth, is that there originally was no significant oxygen. It was mostly methane, hydrogen, ammonia, and water gas. So if water was a gas, what's your intuition about the temperature of that atmosphere? Very hot. Exactly, it was very hot. But the process with time, if we have something that's very hot on the stove and we turn off the stove, does that get hotter or colder? Colder. Right, there's dissipation of heat. So the Earth is the same. We're talking about material things, material objects. So little by little, the Earth is cooling, especially at the crust. Because the crust also, the more crust accumulates, the more it insulates from the magma, from the hot magma underneath. So the outer layer of the crust becomes increasingly cooler and cooler. What happens then that this water that was originally water gas coming from the inside or being trapped, actually the most recent theory is that the water is you can think of the Earth as a sponge was moving through the universe. It was actually collecting water molecules from the universe. Okay, the bulk of the water of the universe is thought to be, of the Earth is thought to be extraterrestrial water, which is amazing. This water molecules, imagine, that makes up all the oceans, all the water on the Earth, which is a very abundant molecule, right? Anyway, that started to condense with the cooling of the crust, the outside of the crust, right? What we call the surface of the Earth. And as it condensed, it started going into the lower basins of the crust, which we call the ocean floor. And there was one kind of bulging thing on one side of the crust, which would have been Pangaea. Well, it's estimated that there were perhaps several Pangaeas back and forth, cracking and reshaping at the other end as this crust was in a fluid state, but before there was any life. At any rate, at some point, we also get liquid water. But these gases remain. There's methane, there's hydrogen and ammonia, which is toxic for today's animals and plants, right? So that is known as a reduced atmosphere because it doesn't have the oxygen. Somehow these cyanobacteria started evolving from other bacteria. So the key issue here is how did they get the process of photosynthesis? How did these pigments became photosynthetic, right? In other words, combining the CO2 that was spewing out of volcanoes, for example, and the water that was abundantly available, there must have been some original chemical reaction that combined that CO2 with that water or some precursor to get the beginning of an organic molecule. At any rate, when we go back to the fossil record, we find that about 3.8 billion years ago, there is abundant evidence, or there's evidence of blooming of these cyanobacteria all over the world where there was water, okay? Huge amounts. And so it's estimated that the bulk of the oxygen that we have on Earth to this day, almost 4 billion years later, was produced by these cyanobacteria, okay, through the process of photosynthesis. So very significant. We have descendants of those cyanobacteria today living, and they can cause those blooms if conditions are just right. They will take off. In principle, bacteria will reproduce every 20 minutes under ideal conditions, will duplicate. So again, we can do that math, you know, every 20 minutes from one bacterium, a single bacterium, and we get that in cultures, right, in culture dishes when we isolate the bacteria to identify what strain it is. One bacterium in 20 minutes under ideal conditions, ideal nutrients and temperature and pressure and all that, will double. So we get from 1 to 2. In 40 minutes, from 2 we get 4, and in 60 minutes, right? In 1 hour, we get 8 bacteria. But then every 20 minutes, it duplicates again and again and again. Within 24 hours, you have billions of bacteria because every population is duplicating ideally, all right? Until it overgrows and they eat up all the culture medium and it becomes a mess. You have to stop the growth at some point before they overwhelm the system, okay? But under ideal conditions, they double every 20 minutes. And so these algae blooms that happen in nature even this day are astonishing, and because the conditions were ideal for some time, and then they end up consuming the nutrients there, and then it goes the opposite typically because they're also metabolizing, because they're alive. They're metabolizing, meaning that they're also respiring, they're using oxygen, not all of the oxygen, but some of the oxygen produced, especially the oxygen in the water. Then that region of water becomes anaerobic or not sufficient oxygen for the other animals that live there, especially fish, and the fish literally suffocate to death because they don't get enough oxygen from the water where the bacteria are. That's why those algae blooms kill all the fish, and then you see the fish washing up on shore, the dead fish, because the bacteria, in addition to producing oxygen that was going to the atmosphere, they sucked up the oxygen in the water. Okay, and therefore the fish, which most of them cannot breathe from the atmosphere, and they suffocated to death. So you see a transition here. The next step from the Archaea and bacteria-like organisms to now cyanobacteria photosynthesizing, but we're still in the prokaryotes. At some point, there was compartmentalization. In other words, other membranes started, or the membrane started to, here's an example of a bacterium that the cellular membrane folds in, and you can see it here very graphically, okay, how the cellular membrane folds in and generates these pockets, all right, these compartments, which could be the beginning of a compartmentalization, meaning specialization or division of labor within the cell itself, all right? And since the nuclear membrane is also a double membrane, this could be a hypothesis of how the nucleus actually originated if one of these compartments essentially engulfed the DNA concentration, the main concentration of DNA, the plasmids of the bacteria DNA. That could have been the precursor. I mean, you can see it here very graphically, the double membrane, the precursor of a nucleus. So one theory. The other one is by engulfing another bacterium that also had a double membrane. Okay. So this would have been the passage from prokaryote to eukaryote, is the engulfing of the DNA material, the genetic material, into a membrane of its own, into a pocket of its own. And that's why this process is called endosymbiosis, which is a membrane within a membrane. I had a little video of endosymbiosis at the synthetic level. I'll hold it until the second part of the class. Basically, I'm showing you evidence here of a process that could have happened based on the organisms that we see today, that we see living today. Some of them can live in streaming conditions, prokaryotes, okay, which would be the primitive atmosphere. Others can photosynthesize, still being bacteria, being prokaryotes, and can photosynthesize, which would have made the bulk of the oxygen on Earth, changing the atmosphere to an oxidizing atmosphere as opposed to a reducing atmosphere. And others could have started membranes within membranes of their cells, leading eventually to compartmentalization of metabolic processes, and especially of protecting the inheritance material, which would be the nuclear membrane. Okay, so these are just idealized diagrams of cells, of animal cell and plant cell. And the main organelles that have a membrane, a double membrane, are the nucleus, the mitochondria, and the chloroplast. Okay. So these membranes, these organelles that have different functions within the cell have a double membrane, just like the cell membrane. And so the nucleus, which is present on both animal and plant cells, the nucleus is surrounded by this double membrane, the nuclear membrane, that makes them eukaryotes. Eu meaning true or real nucleus, a real nucleus. Karyo is again the reference to nucleus. So a true or real nucleus encased in a membrane protected by membrane. So that's the nuclear membrane, which is both in animal and plant cells. And also in both is the mitochondria, which is the energy factory of the cell, right? It recharges ATP at the molecular level. It is in charge of recharging the energy molecules of the cell so that that energy can be used for metabolism, for cellular metabolism. And therefore, since the animal cells are alive, they metabolize, they have mitochondria. And plant cells are also alive and they also metabolize, so they also have mitochondria, okay? And then in addition, plant cells have another organelle that animal cells don't have, and that is the chloroplast, the chloroplast. And the chloroplast is a reference to color. Chloro is a reference to color. And this is the photosynthetic material that we know as the photosynthetic pigments in the chloroplast that are responsible functionally for making that photosynthetic reaction of combining water and carbon dioxide into glucose, and then a side product of that is, again, oxygen. Okay, this little video shows a little bit of these endosymbiosis. It's only a couple of minutes. Let's take a look.
The eukaryotic internal membrane system, called the endoplasmic reticulum, and the nuclear
Sorry, I'm trying to do full screen here. I don't know what happened.
envelope may have evolved from infolding of the plasma.
Oh, sorry. Full screen is not working. It's stuck halfway. Yes, let me try that. Yeah, but how do I get out of it? It's not reacting. It's
membrane in an end.
Okay, hold on. Let me see if I can end Yeah, stats for nerds, that's what I need. Maybe here. Oh, okay. Okay. Let's try that.
Eukaryotic internal membrane system.
Sorry. I'm sorry about this.
Called the endoplasmic.
How do we get rid of the sign now? Show controls. Oh, complicated. Something that is not supposed to be complicated. Let's try that. Okay, we'll keep it that size. We'll keep it that size.
The eukaryotic internal membrane system, called the endoplasmic reticulum, and the nuclear envelope may...
Maybe it's corrupt. You're seeing the same thing that I'm seeing.
Have evolved from...
I go back here, maybe. Try that.
The eukaryotic internal membrane system, called the endoplasmic reticulum, and the nuclear envelope.
Sorry, folks, this is not working. Let me try to go back to the lecture. Okay, I can show this film again when we talk about the other aspect of the beginning of life. All right, I have it also in a Word, so maybe the link is a little corrupt. So the origin of eukaryotes is thought to be by this process of endosymbiosis. So again, another significant step in the evolution of organisms, of living organisms, from a disorganized no true nucleus in the case of Archaea and bacteria, to an organized nucleus protected by a second layer of membrane, which is the nuclear membrane, the eukaryotes. All right, so now we are doing this. We're going into the eukaryotes. So this aspect of the branching here. Well, what we find in the eukaryotes is a great variety of species, a great variety of species. And classically, these were mostly microscopic, and they were grouped as protists, but the protist is really a grab bag. It doesn't have much phylogenetic consistency to it. They're very different organisms. Okay. And I was looking at here, for example, where we are with Meyer. Yeah, the prokaryotes. It's a very heterogeneous group. Most of them are unicellular or colonial, meaning some kind of a conglomerate association of cells that may begin to specialize a little bit. Some are producers, some are consumers. In other words, some photosynthesize, others don't. For example, the Euglena, which moves around like an animal, has a flagellum, but at the same time it photosynthesizes. Some are freshwater, some are saltwater. Some are motile, meaning that they move around. Others are sessile, meaning that they're anchored, stuck to the bottom somewhere. But by and large, they're all aquatic. And the fact that they're aquatic is probably the only factor that they have in common for all of the protists. And it also points to an early primitive beginning, early on in evolution. And it's put around 2.7 billion years ago, meaning that the cyanobacteria spent about a billion years, because, oh yes, all these protists, they all metabolize and they all use oxygen, okay? They'll use oxygen for the metabolism. It's another characteristic. So it's estimated that the cyanobacteria were producing oxygen for about 1 billion years. Okay, from about approximately, if we round it up to the nearest whole numbers here, from 8 billion years to 7 billion years ago, which is when these eukaryotic metabolism species began to emerge, estimated. For 1 billion years, these cyanobacteria were producing oxygen on the Earth. It's almost maybe about 20% of the age of the Earth, right? And that's how come we have actually so much oxygen today. All right, let's see where we are then. Here are some examples. Many of these are microscopic, some are individual, some are colonial, but some are macroscopic, like the algae we're placing there. These, you recognize this algae, this alga? Miami Beach, yes, sargassum, the seaweed. Yeah, smells when you get to the shore and all that. But this is actually a whole condominium, be surprised. Next time you're at the beach, if you see these floating around, which is actually called flotsam, lift it up, raise it, and shake a little bit like that. You'll see all kinds of little crabs and mostly shrimp jumping out, but I've seen some fish also sometimes. And it's a whole condominium of an ecosystem, right? Yeah. And they get protection in there. And many of those organisms, many of those animals have a camouflage that looks more or less like the algae. They'll have the coloration and the pattern. And there's one particular fish, histrio, the sargassum fish. I don't know if you've ever seen those. Those are fascinating. They're very voracious. They can actually eat another fish almost their same size because the lower jaw comes out into a huge gape. Histrio, see? Look how similar it is to the actual sargassum weed. And the fins are super adapted, especially the lateral fins and the pectoral fins. They have these fin rays that crawl, that are articulated, and they can crawl through the sargassum. So they mostly crawl more than swim. They're kind of awkward swimming. But they are very good, very dexterous at crawling. All right, so that's a super adapted fin. Some can get pretty big. Okay, anyway, back to the other species. This is a rheophyte. This is a red algae, also marine. And they form colonies, sometimes primitive colonies. These are the quanoflagellates that we saw on the break with the animals. These are volvox. It's a little ball of green algae. There's our individual cells within, so you can see the beginning of a colony here, or cellular specialization. What would happen with colonies, for example, it's the beginning of multicellularity. It goes through a colonial stage, right? The beginning of multicellularity goes through this colonial stage. What happens is, as the colonies begin to form, colonies of cells, of individual cells, working together, over time, they may start specializing to doing, let's say, digestion or nutrition. Other cells may be formed in anchoring the organism to the bottom. Okay, other cells may begin to specialize as a defense mechanism. And you end up really with a colonial organism, which to this day, this exists. The Portuguese man of war, you'd know it if you get stung by it. Ever get stung by Portuguese man of war? They are nasty. They are from and they're related to the salenterates, they're related to the sea anemones and coral, all right? And this is actually considered a colonial organism. that has, for example, the surface part, the top part, the floater. These are the cells that form the floater, and inside, this is actually, I believe it's CO2 that accumulates inside the balloon. Is it CO2? I think, yeah, and it floats, right? But these are specialized cells to make the float within the colony. Other cells form the mass of tentacles that hang down, but the mass of tentacles here, their cells are closer to the body and connect to the floater here. These cells have an additional function of digestion, of digesting the prey. And it's kind of an external digestion. They excrete the digestive enzymes, and then they reabsorb the nutrients, the product of digestion. These tentacles can contract or expand. Some of them can expand 10, 15, 20 feet length. So you can see the floater out there and say, oh, no problem, that manowar is far away. Yeah, but if the current is coming toward us, then those tentacles could be coming this way, and we feel it. Typically for salenterates, the poison is a neurotoxin, it's a neuroparalyzer. They paralyze, they tend to paralyze the prey and digest the prey paralyzed but live. So they like fresh prey. Okay. And then another layer of complexity. So this is really a colonial organism that is formed by these groups of specialized cells. Some are tentacles, some are floaters, some are digestive cells, and so forth. And you know what loves to eat this, as poisonous as they are? The turtles, the sea turtles. Sea turtles, they love this mass, especially, yes. And so I've seen photographs of sea turtles that are chomping on the Portuguese manowar. And of course, the carapace protects them, the shell, and because they're reptiles, right? So they have scales, and that protects them. But in between the scale, there's skin. And so, for example, their eyes are bulging out like that because they're getting all stung but they love to eat these things. Some species do. And so they pay the price. You know, it's like the hot food for Mexico and India, the curry. The first thing that happens with curry is that it's like a water spout on the top of the head. You start profusing this water, the sweat from the top, and then it covers the whole body. Right? It opens up all the pores. Can't stay away from that curry. And in some animals, actually, there's a secondary adaptation that happens with some animals that eat these salenterates, these stinging cells that, let me see if I remember my marine biology, what are they called? The choanocytes, I think they're called the choanocytes. These stinging cells get incorporated into the tissue of the animal that is eating them. And then if that animal is touched at the skin level, then those choanocytes will fire out. It's like a secondary adaptation. They get a secondary protection from eating these or fooling around with these salenterates. Okay? But it's a secondary adaptation. At any rate, the precursors of multicellularity, which is thought to have happened many times in evolution, the intermediate step there could be colonial cells coming together and specializing division of labor. So where are we? Let's see. Oh yes, the preconditions for moving forward in life are how to acquire energy, right? And how to replicate. So the two main functions of all living organisms are going to be metabolism and reproduction. So if an organism or a thing that we encounter crawling around, if that thing is metabolizing and is capable also of reproducing, generating another one like itself, then most likely it's a living thing and it's not a robot that is crawling around, okay? All right, so the eukaryotes then is this fusion of cells to get especially the three main or initial organelles, right? The nucleus, the mitochondria, and the chloroplast. What happens? We're still with soft tissue. So the fossil record, the skeptics are going to ask, well, show me the fossil. However, the fossil, the most we can point at is those stromatophytes, because this is all soft tissue. So it's not being really preserved, okay? The paucity of the fossil record. So the protists, I already talked about that, a very heterogeneous group. It has all those characteristics. But now multicellularity is getting more and more sophisticated. When cells begin to specialize within an individual, within an organism, that division of labor allows for overall better living. Larger size, longer life, less dependent on particular environments, more resistant to environmental change. All those characteristics that come with multicellularity and division of labor, because also with multicellularity and division of labor, now we have the possibility of going into tissues, specialized tissues. And then tissues that have similar function come together to form organs. Organs with similar function, again, will make systems, organ systems. But then there's a jump to have the entire organism. We need systems that have different functions, and they can be complementary functions. But at the level of system, you know, the respiratory system does not do what the digestive system does, which does not do what the nervous system does, and so forth. Because I can lay out the dozen systems of our body here intact, but I don't have a whole organism, I don't have a functional organism, because they have to be integrated. And those systems are diverse. However, when we go into the systems themselves, we find similarities. We find at least those three layers of complexity. From cells with similar function forming tissues, tissues with similar function forming organs, and organs with similar function forming systems. Okay? All this possible by a greater and greater cellular differentiation or specialization, so that the liver cells would not do what the brain cells do, and so on and so forth. Understand? And that also is how we go from totipotent cells in the early embryo, meaning the zygote and the morula. We'll see it in more detail next semester, all the way to fully differentiated cells, from totipotent, pluripotent, multipotent, and finally fully differentiated. That is the differentiation pathway of the vast majority of our cells in the body. So there is an ontogeny and phylogeny, if you will, there in the broad sense, at that level of going through the layers from totipotent to fully differentiated cells. So the divergence is not only internal with division of labor, if you will, specialization, but also at the level of the variety of species that develop, okay? So the variety of species is radiating out. In evolution, there is a tendency to diversify because of niche specialization. In other words, some organisms are going to specialize in living in the ocean, at the bottom of the ocean. Others will specialize living in the water column. Others toward the surface. Others will make the transition into land, and their niche specialization then is leading them to greater and greater radiation into a variety of species. Eventually, we end up with the three large kingdoms that I was proposing for a simplified phylogenetic tree of plants, animals, and fungi, okay, the simplified version. So we see that these three branches are already existing in the Precambrian, and take off. They take off in large size with the Cambrian explosion, which is known as a radiation. And this happened around half a billion years ago. So now we're into the scale of millions of years. Okay. Let's go back to the slides then. We get this proliferation into the three main kingdoms in the simplified version of the phylogenetic tree: plants, fungi, and animals. And a great variety of size and specialization and so forth. For example, from mosses and lichens and ferns and so forth, small, rather primitive, to flowering plants, which are the most evolved of the plants, and the largest also, the flowering plants. Okay, where the actual flower is a super adapted cluster of leaves, of former leaves, that now become the reproductive organ of the plant itself. So you understand that a leaf is not a reproductive organ. In principle, it's a photosynthetic organ. But flowers, which are considered to be super specialized leaves, and that's why flowers also have color, because they have the pigmentation, the photosynthetic pigmentations, right? Those are reproductive organs of the plants. So it's a super specialization. You see where then these organisms have metabolism and they have reproductive capability. So they qualify as living, if you will, with those two main characteristics. In contrast to the plants that are producers, that they produce their own food and the food for the rest of the living world, are fungi, which are not producers, even though they tend to look like producers somehow. You know, an anemic producer, an anemic plant. No, they don't really have photosynthetic pigments. So if they're not producers of food, of glucose, then they have to be consumers. But they are a specialized type of consumer. What kind of consumer are they? They are saprophytes, right? In other words, let me put it in here, in parentheses. What do they love? The phyte is a reference to love. They love decaying material, saprol. They love what's rotting. Okay, so they feed on rotting, decaying organic material. Those are saprophytes. And they can be microscopic, like yeast, for example, which is a type of fungus, right? The yeast that rises the bread. What's the yeast doing? The yeast is feeding on the bread, is breaking down the starch into glucose, and is producing CO2 in the process of metabolizing. And that CO2, carbon dioxide, is a gas, naturally, and it bubbles inside the bread, and that makes the holes in the bread, okay, which then diffuses out. Two very large, as you can see, this is an exceptionally large fungus, right, that this man is carrying. But most fungi don't get that large, but there are some fungi that get huge underneath, especially with the what is it, the myceli, or the it's not a true root system, the hyphae, the cells that expand sometimes a huge amount of territory underneath the soil, all right? Dozens and dozens of meters of the hyphae, which are not true roots. They act as roots, but they're absorbing nutrients. They're not absorbing, they also absorb water, but not for photosynthesis, for nutrition. In the animal world, also from microscopic worms all the way to the largest animals on Earth, the cetaceans, or whales and dolphins and orcas, which precursors, ancestors started in the ocean, aquatic life, migrated to land, and then secondarily remigrated back to the ocean, and that's why they're mammals. They have lungs, they don't have gills. The nostril migrated to the top of the head here, and it's the blowhole, right? And all these mammals have to come up to the surface to breathe, and they can spend maybe several hours underwater, but at some point they have to come up to the surface. Mm-hmm. Let's see, what else do we have here from Meyer? We're down to the vertebrates, yes. Again, about half a million years, 500 million years ago, about half a billion years ago, coinciding with the Cambrian Explosion, which we'll see hopefully a little film about it in the second part. Once we get vertebrates or chordates, which is actually the, I should put here, chordates. And in parentheses, actually, because chordate is a notochord, a swimming chord, a semi-rigid structure along the dorsal side of the individual. Oops. But these arrows got discombobulated. Sorry. Okay. Let me put the little arrows back. From reptiles, birds, and mammals. Yeah. What I was saying is that the chordate is a precursor to the vertebrate because a further specialization of the chord, of the notochord, of the swimming chord, is that it becomes fragmented to give it more elasticity. Okay, and that's the vertebral part. At any rate, these would be the precursors to fish, and then some primitive fish eventually started migrated into land, and descendants of those primitive fish became more and more adapted to land, so that especially the lateral fins and the pectoral fins were used for crawling, and the digits, the fin rays started segmenting for articulation and so forth, so that we get true digits, and eventually the tetrapods, and the amphibians, even the amphibians of today, you know, the larval stage is aquatic, typically, the tadpole, right? Whereas the adult stage is land, tetrapod, frog, or the toad. Eventually, ancestors of these amphibians branched off and developed scales, and then ancestors, so pointing toward the reptiles, where they can hold on to moisture, because the problem with the amphibians is that their skin is exposed. They don't have true scales or hairs, so they tend to dehydrate, and that's why they need to be close to water or a humid environment. They're on land, but they're limited. Whereas the development of scale, which is basically keratin protein, is all over the place, right? And it's a consolidation of keratin protein in clusters. This is the precursor of a scale. Just like the hoof, the hair, scale, that's all keratin protein, all right? Eventually, those would be the precursors of reptiles that get to have scales proper, which means that now they are more isolated from dehydration, and they can be on land better in more extensive territory. Further sophistication of that keratin protein is to become more and more specialized into a hair as opposed to a scale. And the branching, whether the hair happens on terrestrial animals, would be the mammals, but also some of these other ancestors developed very sophisticated hairs that we call feathers today, And so that gave them the ability also to take off and fly. And also their bones became hollow and more lightweight, other adaptations for the birds. But it's estimated today that both birds and mammals diverged from some primitive reptile ancestor to the current. Remember, there are no contemporary ancestors, right? So these are all ancestors to the contemporary animals that we see today. So that's just an example of radiation there of a species, all from a common chordate originally. So again, to the branching effect, the phylogenetic tree with some simplified layers. From the Precambrian to the Cambrian is what is known as the Cambrian Explosion. You can see here prokaryotes, again bacteria and archaea. Then the branching to eukaryotes. Of course, the prokaryotes have to branch out there. All right, and because we have prokaryotes today, we have bacteria and eukarya living today, right? So they had to branch out before the true nucleus developed. Moving forward, at some point, the multicellularity from colonial to actual tissues and organs. Oh, thank you very much, John. Thank you, appreciate it. Okay, so another branching just before the Cambrian, but then when we go into the Cambrian era, then there is it's called an explosion or radiation, diversification, a great diversification, the metazoans, all the larger animals and plants that we see today, but especially on the animals, marine animals, okay? Further branching on today to the 2 or 3 dozen phyla that we have of living animals. If you want to look at it in a more sophisticated way, here you go. Here it shows about a dozen major phyla of the animals. We are over here in this branching of the, here with the moose. What is moose? Mouse, Mus musculus is the common mouse, and Homo. So this is a line of mammalian here. You can go further back. As you see, the nodes are indicated by numbers. Okay. You can trace it all the way back. At each one of these nodes, there is a significant event happening for the organisms, and they coincide more or less with these epochs, which would be a change in the environment. But you can see here is the Cambrian. You can see how many more lines, phylogenetic lines, branch out during the Cambrian and then pretty much stabilize after that, compared to fewer branching before the Cambrian, okay? Fewer nodes, fewer branching, but then this, what is known as the Cambrian explosion. What do I have next? All right, before we go there, let me see for Meyer. Yeah, okay, so he ends there. Let me finish with this because I think we all need to get out of this polarizing here. I'm sorry about the temperatures. So the original theory of how life came about on Earth was known as the spontaneous generation, the theory of spontaneous generation. In the second half, I'll show you a few more little videos that explain this in more detail, but basically the idea of spontaneous generation, that life emerged just from rotting meat, for example, or just by the similarity of shape, from non-living things to living, That theory is totally disproven today, and yet it was the prevailing theory for many years, for a century, and replaced today with this theory of descent with modification, which is just a synthetic way of talking about evolution, right? Descent with modification, and this would be the mechanism, would be putting together Charles Darwin's theory of evolution with Gregor Mendel theory of inheritance factors, right? What is known again as the modern synthesis from Haldane and others, Fisher at the beginning of last century. So 2 centuries ago, in the 1800s, with Darwin and Mendel primarily, and then last century, the beginning of last century, with Fisher and Haldane and others, Meyer included, in the modern synthesis, is the prevailing theory today. All right, let's stop there. We can look at the videos when we come back and thaw out a little bit. Any questions or comments? No? Okay, we're gonna stop here. So let me pause the recording. Okay, folks, welcome back into the second half of this lecture on the origin of life on Earth. And I'm going to be covering this handout here. It's based on Barton and colleagues, a book on evolution from 2007. And these are 7 steps for the possibility of generating life starting from inorganic material. Precursors. In other words, try to simulate in the lab the primitive conditions of the atmosphere. Sorry, Father Jose, may I ask you to mute your microphone if you can? But if you have a question, turn it on so that we can hear it. Otherwise, we just hear the background noise in your room or wherever you are.
Okay, thank you.
Sure, if you can mute the microphone, but if you have a question, turn it on, and then, or a comment, all right?
Okay, thank you.
You're welcome. Thank you. All right, so let's see. Yes, in 7 steps, possibly, life could be generated synthetically in the lab from inorganic material, trying to mimic again the primitive atmosphere and so forth. So we're going to go through these step by step. All right. Again, he has at the beginning of this chapter in Barton, he again confirms the beginning of the Earth with planetological evidence and radiocarbon dating and so forth, 4.5 billion years ago. Then organic life, again, around 3.8 to 4.2 with more recent discoveries. And then he goes into the 7 steps that we're going to cover. Something here. Oh, yes, so we had left off with this idea of spontaneous generation. This is the contrast that I want to show. It's another little handout here. This is the videos. I guess I left it embedded here, so let's try this. Oh. There we go. Let's see if this link is going to behave better.
Prior to Charles Darwin.
Oh, still getting this, so it's obviously not the video. Let me try moving it. Yes?
Yeah.
Let me, because this is where I got the links from. Let me try the, I don't know if this makes a difference. Just, let me go to the beginning. Give me a minute here. As you can tell, I'm not techie, but by hook or crook, whenever, for some reason, whenever I put the links here, they tend to work. Oh, you see, have become inactivated. Let's try that. Maybe yes, maybe not. This doesn't work, we'll go to YouTube. No. Okay. Doctor, what's your suggestion? Go to YouTube?
Go to YouTube, yeah.
Okay.
And I'm not technologically.
Okay. Now between all of us, we'll put it together. Okay, I'm in YouTube now. So put it in this search. Okay, let's try that. Oh, okay. From here. It is that
Prior
video.
to Charles Darwin,
I don't know why. Anybody have any ideas why this is happening? Let me pause recording for a moment so we can figure out this. Okay, folks, very sorry. We got the video going. So here we go. 2 minutes. The longest 2 minutes in history.
prior to Charles Darwin, scientists had some rather strange ideas concerning how life began. They believed that living organisms came into being rapidly and spontaneously over a period of just a few weeks. The scientific community believed in spontaneous generation for 2,000 years. And it is a stark reminder that even the majority of scientists can be wrong. In his classic 17th century description of spontaneous generation, scientist Jan Baptist von Helmont suggested that mice came from dirty underwear.
Wanneer je een stuk gedragen ondergoed, if you put a piece of sweaty underwear in an open-mouthed jar, together with a piece of wheat, after 21 days, the ferment coming out of the underwear changed the wheat into mice. But what is even more amazing is that the mice are not small or aborted mice, but adult mice emerge.
Another evidence offered for spontaneous generation was the rotting meat experiment. 17th century scientists observed that if meat was placed in an open jar, maggots formed on the meat weeks later. They conjectured that life, in the form of maggots, arose spontaneously from rotting meat. But in 1668, Francesco Redi, an Italian physician and scientist, overturned this idea. He suggested this proof of spontaneous generation was nothing more than contamination of the meat by flies. When flies landed on the rotting meat, they laid their eggs. Over time, these eggs grew into maggots. Later, the maggots changed into flies. When scientist Redi prevented flies from landing on the meat with a piece of cheesecloth, maggots never formed.
The beginning of scientific experimentation, right? Observation and experimentation, right? It's a transition. All right, so this is a good segue for getting into this possibility of generating life in the lab. Let's see, this one here. So now we're going to use this procedure. Again, beginning with a reduced atmosphere, right? Reduced atmosphere, meaning where there is no oxygen, but there is methane, there's ammonia, there is hydrogen and water vapor, which are all products, and some carbon dioxide also. It's all product of, say, volcanic activity and geologic activity of the Earth. Combining these molecules to see if one can get the beginning, the precursor of life, which is, inorganic molecules into organic. That was an experiment done by Miller and Urey, the famous Urey-Miller experiment, or Miller-Urey experiment, back in the 50s. And there's a little film for that that explains it very well. Here we go. Okay, I'll send you this sheet of, I sent it, yeah, I sent you the links already in an attachment, but we're trying Firefox, right?
In the 1920s, Oparin and Haldane hypothesized that the atmosphere of the primitive Earth contained gases such as nitrogen, ammonia, methane, carbon dioxide, water vapor, and hydrogen. Oxygen did not become a part of the atmosphere until the advent of photosynthesis millions of years later. It was further hypothesized that under these conditions, organic molecules could be formed if there were a source of energy, such as electricity from storms or ultraviolet radiation from the sun. In 1953, Stanley Miller and Harold Urey tested this hypothesis. They constructed a closed system with a reservoir for water, sampling ports, a chamber for the input of electrical energy, and a condenser to convert the water vapor back to a liquid. The researchers then added water, hydrogen gas, methane, carbon dioxide, ammonia, and nitrogen. The water was heated to form a vapor, and electrical sparks were generated to mimic lightning. This apparatus simulated the possible conditions of the early Earth. When the water was analyzed after 1 week, they found simple carbon compounds such as formaldehyde and hydrogen cyanide. These compounds then combined to produce formic acid, urea, and amino acids. In similar experiments carried out over the next several years, various organic compounds have been synthesized. These experiments showed that simple organic compounds, including the building blocks of proteins and other macromolecules, could be formed from gases present in the primitive atmosphere with the input of electrical energy.
Right, so you see, this experiment was very important back in the 50s to prove the possibility of going from inorganic molecules to organic molecules. Was the first step. Now, this is not life yet, but at least we have the basic building blocks for organic molecules, for the macromolecules. And again, the macromolecules, the organic molecules, are four, and they are four groups. Let me see where I'm going to put this. No, on this. Let me put it on this handout. I'll send you the updated list, the updated handout by email, okay? So, four groups of organic molecules. And you see how significant these are, how very important. These are also known as macromolecules, simply because, what's your intuition? Why are they called macromolecules? Yes, they contain, exactly, they contain millions of atoms, thousands or millions of atoms, in contrast to normal small molecules like water only contains 3 atoms, right? CO2, 3 atoms, etc. So these are, is that large enough? You can go a little larger. So proteins, lipids, carbohydrates, and amino acids, which are DNA and RNA. These are the 4 groups of macromolecules. So this is the beginning of a new, of the possibility of life. Where is that? What to change? Okay, here. Got them all in a group. So this experiment in the 50s has established that, and this experiment has been repeated and tweaked and adapted many times over the past 40, 50 years, the Miller-Urey experiment, but very famous. You can see the apparatus is around there. There are all kinds of designs for the apparatus that is around, okay? All right, next step, once we have these macromolecules, is what is known as a chemical evolution. Before a biological evolution, chemical evolution. The discovery that molecules also, chemical molecules, have a dynamism that they can react with each other, all right? And they can produce products by chemical reaction. Chemicals, and especially when you have molecules aggregating, coming together, polymerizing, for example, forming chains, all right? They can self-assemble under certain conditions, especially if there's ultraviolet involved, because ultraviolet can generate links between molecules and form what is known as a polymer, a chain of the same basic molecule repeated many times. And that begins to give structure to these macromolecules in a larger, more complex way. These structures then could develop a 3-dimensional shape that can have some impact, the 3-dimensional shape, because of the particular shape. For example, proteins have one area of the protein that is the active site. Enzymes are proteins, and they work what is known by the lock and key example, how the teeth of the key manage to open a lock, but not every key opens every lock, all right? So it's pretty much a 3-dimensional shape issue. This can happen by polymerization of smaller subunits. That would be the second step, which overall is known as chemical evolution, from simple organic molecules to form more complex ones. And this could become a primitive network of macromolecules. I think I had another little video here of polymerization, which is interesting for nylon. And these are short, here, polymerization. It's a little older video, you can tell just from the setting, but the main point here is that these polymers can be formed at the interface.
Polymers are found in nature.
What did I do wrong? I went back to, well, I have to go back to Mozilla, right? Let's try that. Oh, I should. Changes. So tedious, but if it gets us there, it's a good thing.
Polymers are found in nature. Spider silk is one of the strongest of the natural polymers.
Spider silk.
Chemists have always sought to duplicate what is found in nature, and especially in this process called polymerization. An example of that would be to take these paper clips and allow each one to represent a molecule called a monomer. We can then link these monomers together in a chain to form what is called a polymer. We can see that process happening if we take these paper clips and gradually add them to our reaction vessel, this box. Closing the vessel and carrying out the chemical reaction, usually with the aid of a catalyst, we can then remove the polymer, a synthetic fiber. But what you're interested in seeing would be the real chemistry, and one of the most useful of the polymers today that we find is nylon. Here to this chemical, we're adding a few drops of phenolphthalein indicator. It's basic, and therefore the color will allow you to see this layer. We're going to pour in a second layer of another chemical. The two chemicals form two separate layers. If we reach in...
Sorry. Okay, this is a crucial point here. So you notice these two layers could be formed maybe in an area in a shallow surface, or maybe the deep ocean. The two layers can be formed by stratification, either temperature or just the two compounds are not soluble in each other. All right, so you can see, you can imagine that these two layers could be formed naturally by some natural phenomena of these two substances accumulating one on top of each other. Okay. Now, watch what happens when he reaches in with a pair of tweezers in between the two layers,
To the interface of those two layers,
the interface,
where the molecules come together and react, we can pull out a synthetic fiber.
That's the polymer.
This synthetic fiber, slightly colored by the phenolphthalein, is nylon. An application of polymerization can even be found in the toy store in the form of string confetti.
From the sublime to the ridiculous. Very good. But you see, this is a synthetic polymer. But nature does that. For example, he mentions in the silk of many insects, the spiders, which have, I think, 6 different types of glands in their abdomen to make different types of silk, and they're mostly proteins, protein polymers, right? Other animals do that too. Anyway, so that would be the second step there. Let me see. There. On the chemical evolution, going from monomers to polymers, right? Which can happen naturally, especially if there's some kind of energy source like ultraviolet radiation. The next step is self-replication. The possibility of splitting or forming aggregates from individual chemical pools, for lack of a better word. There's another little video here of this replication and compartmentalizing with something called quacervates. Ever heard of quacervates? Right, a quacervate is a droplet, a microscopic droplet that can be made up of many different substances, typically organic substances. And they have certain properties that they can, if you change the pH or you change the electric conductivity on it, they will start reacting to the atmospheric change, to the environmental change. Okay? So these little quacervate droplets, by changing the voltage on them, will start moving around and aggregating, clustering, and then breaking up. Very interesting. Again, this is not life, but these would be possible precursors of life in these steps. Okay, this one has no sound, but let me start at the very beginning here. And you'll notice down here, it will give the voltage per second, all right? The voltage that is being conducted through the little strip of the yellow dots. Each one of those dots is a coacervate. It's a micro dot, so it's magnified under a microscope. And they could be droplets of organic material in them. So starting at 10 volts per second, increasing 30, you notice how they start reacting just to the voltage going through them. A little bit of motion, but you'll see that they'll start aggregating at some point. See here, I think this one engulfs another one next to it. See how they're moving and aggregating. 50 volts. See how they engulf each other? It just by changing the voltage is the same substance. These organic substances that have this property. And then when it's greater voltage still, they fragment. See how they're fragmenting now?
Yeah.
Okay. So this could be like the precursor of a reproductive system, very primitive, asexual reproduction for sure, just by binary fission, which happens in the bacteria and the archaea. Okay, so that's another step there of the self-replication and aggregation. Another one, compartmentalizing. Compartmentalizing is within the cell itself, or the protocell, to have different areas or regions of this protocell, all at the microscopic level, that have particular function, Here, the boundaries are important also because what's going to form that bubble? And there are some molecules that are called amphipathic. That's too small. Let me make it a little larger. Amphipathic here, meaning that the molecule has two sides. One side is hydrophilic, it likes water, and the other side is hydrophobic. So when there is an interface between two substances, these molecules will self-assemble, will align, so that the hydrophilic end will go into the water phase, and the hydrophobic end will be away from the water, right? And it has to do with static, hydrostatic, if they're positive or negative or neutral molecules. But some molecules have that property, and proteins have that. In fact, that's how they embed themselves into the membrane of the cell, that there will be a hydrophobic end, which will be into the inside of the membrane, and the hydrophilic will be facing out of the membrane into the medium, whether it's inside the cell or the intracellular medium. So this could eventually form layers or bilayers when there's water on both sides. Hydrophobic end of those proteins we're going to be facing inside, which is really what we have, and the phospholipid bilayer of cells today. I had another little video on compartments. Let me see. Okay, engulfing, this is one of synthetic molecules engulfing another little bubble next to it. They're called protocells. You see here, when it starts, this one is stimulated by a magnetic field, all right? The magnetic field is off, and this bubble here is stay put. But now, when the magnetic field is on, it starts moving, and it starts engulfing these other protocells that are in front of it. These are synthetic. These were generated in the lab. This is not a living cell or anything like that. But you see that it has this property. When it's polarized, it starts moving by itself and engulfing what is in front of it, but only on this anterior side. The back side of this bubble does not engulf. I don't know if you noticed, but also there's a second action undergoing when these bubbles go into the larger bubble. They start to break up. They start to break up and get maybe dissolved or something like that, and start losing their integrity. Okay, so these are all synthetic processes that are pointing toward the possibility of life, generating life originally from inorganic material if they're linked together. The genotype to the phenotype is basically getting a molecule of inheritance or a molecule of memory, which for many decades it was thought to be proteins, simply because of the chain with the polymerization. It was thought that that polymerization could hold information, right? But then it was realized that the early information molecule was most likely RNA. And today we have many different types of RNA. It's number 6 here actually, many different types of RNA that have specialized functions in the cells. And it's thought then that that branching out, that diversification of the RNA is because there was originally an RNA world. In other words, that the original molecule of inheritance was RNA, but it was not stable enough. And the basic principle for holding the information would be the same, which is alphabetic, the base sequence, all right? The base sequence, just like the letters in a word, that how those letters are arranged will make a difference in the word. But the downside of RNA is that, first of all, it's a single strand instead of a double strand, and that makes it unstable because it tends to wind on itself because those bases do tend to pair. Okay, so it winds on itself, what's called hairpinning and all that. And so that could actually stabilize it, and hairpinning RNA could be a precursor for a DNA molecule, which is stable because now the bases are paired. And what's showing on the outside is just the rail of the ladder, of the DNA ladder, of the helix. Okay? And further stabilization is the twisting or the coiling, and then the supercoiling, coiling on top of itself to make a wad, as it were, of concentrated information. And then the very final step, of course, is the predominance of DNA as the molecule of inheritance for all living species. So that must have started very early on in this process of life in order for that DNA to embed itself into all the species that we know today. Again, tracing it back to about 4 billion years ago when life itself started. Okay. So again, the two characteristics, the metabolism and replication or reproduction, or rather metabolism and reproduction, okay, for having a living system, a living organism. So each one of these steps, as I've shown with the videos, has occurred in labs throughout the world, but no one has managed to put all the steps together yet, all right? And to actually start with methane and ammonia and hydrogen gas and put it all the way through to a little living cell over here, an actual living cell, right? As primitive as it may be. Because even the most primitive living cell that we see today has millions of molecules and metabolic pathways that are extremely complicated, right? So the very most simple, it would be what is known as a protobiont or a protocell. That would be very primitive here. Yeah, the word is here. A protobiont or a protocell would be much simpler than the sophisticated cells that we're used to today, simply because this protocell does not have the benefit of 4 billion years of evolution. But all this to say that it can be done in the lab, okay? And therefore, the individual steps, now the big challenge is putting all of those steps together, because each one of these steps takes quite a bit of background experimentation to do. For the rest, what I have actually is on the geologic era, but I'm seeing that it's almost 12:15. So no, 9:50. We go 12:45. No.
Yeah.
Oh, okay.
Yeah,
Oh, okay. Oh, all right. Good. Okay. So we have about half an hour. All right. Good. I was confused with the time. Then let's move forward. Here's another one. Most of these are short, except one that is a little longer because he puts it all together in an explanation. But I'm giving you the several pieces first as a mosaic. And then the scientist, it's one of those TED Talks that is about 15 minutes, more or less, 14, 15 minutes. Okay, this is endosymbiosis. It's another process, essentially, of...
The eukaryotic internal membrane system, called the endoplasmic reticulum, eukaryotic internal membrane system.
Sorry. Okay. This is just a diagram of how it could be explained that from the cell membrane, a nuclear membrane could develop, all right, from the extension of the cell membrane. And there is, in fact, another organelle, a reticular organelle, a mesh-like organelle in the cells that is called the endoplasmic reticulum. And it continues with the cell membrane, as you can see here, all right? And it's a whole network, it's a mesh of fibers, if you will, mostly protein fibers. And that's where the ribosomes are. That's where the protein synthesis occurs on the endoplasmic reticulum. And one theory, the most prevailing theory, is that an extension of that endoplasmic reticulum could eventually wrap around the DNA and form the precursor of a nuclear membrane, which would make that transition from prokaryotes to eukaryotes, which is it's a sophisticated, it's a very relevant transition now when the DNA is protected in a second layer of membranes. It gives much more viability to those types of cells. Okay, so here we go again.
Called the eukaryotic internal membrane system, called the endoplasmic reticulum, and the nuclear envelope may have evolved from infoldings of the plasma membrane in an ancestral prokaryotic cell. Such infoldings are common in modern prokaryotic cells. The theory of endosymbiosis proposes that a critical stage in the evolution of eukaryotic cells involved endosymbiotic relationships with prokaryotic organisms. Microorganisms that live within other cells and perform specific functions for their host cells are called endosymbionts. According to the theory, energy-producing bacteria may have been engulfed by a larger primitive cell and come to reside within it, eventually evolving into what we now know as mitochondria. Photosynthetic bacteria use...
Sorry, let me, I've been too clear. You can see the double membrane here, right? This is an electron micrograph in the scale of nanometers. Okay. Sí. Perfecto. Okay, I'll take a look. Thank you, Henry. Thank you very much. So you can see the outer membrane is rather flat, if you will, continuous, but the inner membrane folds in and out, okay? What do you think is the function? Again, we're looking at structure, but functionally, why would that inner membrane fold in and out? What's happening? Give you a hint, is analogous to the sulci, right? It's called sulci, the infolding of the brain of the gray matter of the surface of the brain, the convolution. Surface. Exactly, it's a larger surface because in the same mass you have it's convoluted, it's an infolding, and you can spread that out, and it covers a much wider area because the action, the metabolism is happening along the surface of the membrane. In this case, the inner membrane of the mitochondria, that's where the ATPs are being recharged. So it needs more surface, okay? So nature is repetitive that way. If something works in one system, it will continue to make more of the same. All right, so that's for mitochondria.
Photosynthetic bacteria use photosynthetic pigments embedded in internal membranes to derive energy from sunlight. These bacteria may have come to live within early eukaryotic cells, leading to the evolution of chloroplasts.
Like, for example, the cyanobacteria. This could have been a cyanobacterium type that got engulfed and survived the engulfment, was not phagocytized.
Several facts provide evidence for the endosymbiotic hypothesis. A few examples include: 1. Mitochondria and chloroplasts contain their own circular DNA, similar to DNA in bacteria. 2. Both mitochondria and chloroplasts are surrounded by two membranes. The inner membrane is probably evolved from the plasma membrane of the engulfed prokaryote, while the outer membrane is probably derived from the cell membrane of the host. 3. Mitochondria are about the same size as bacteria. 4. Mitochondria appear to have been derived from purple bacteria, and chloroplasts derived from photosynthetic bacteria.
Okay. So that's with regards to endosymbiosis, which is the prevailing theory of how we end up with these organelles inside the cell, particularly the mitochondria and the chloroplast. Of course, we as consumers, we only have mitochondria, but the plants, in addition to mitochondria, they also have the chloroplast for photosynthesis. Oh, this one on the hydrothermal vents is also an interesting one, because this can lead to the proper environment. The proper environment where these chemical reactions may happen. Okay. Let's try this other one. Have you ever heard of these hydrothermal vents? Anyone? Yes, okay. This is in the deep ocean floor where typically volcanoes are going to emerge, or there are cracks actually in the crust, and then some of that water hits the magma, or there is material coming out from the lower layers of the crust into the ocean water, and of course that chemical is extremely toxic, is hot, very hot, but that has created a whole cycle of life in the deep ocean floor where there is no sunlight. The sunlight is totally filtered. And so it's a different process. Instead of photosynthesis, it's going to be chemosynthesis. In other words, using mostly sulfur and other chemicals from the environment to produce energy. All right, chemosynthesis is an alternative to photosynthesis in these deep ocean vents.
So right now we're at a depth of 3,275 meters, looking at an incredible black smoker hydrothermal vent chimney. That black fluid coming out there is likely.
Excuse me. So 3,000 meters, that's 3 kilometers. That's about 2 miles down. Okay, 2 miles deep.
Between 300 and 400 degrees centigrade. These ecosystems are literally islands or
300 to 400 degrees centigrade is the temperature of the stuff that is oozing out. The average temperature of the ocean depth is about 4 degrees above 4 degrees centigrade because it doesn't freeze, right? Icebergs form down there, but the water is still liquid. So it's around 4 degrees more or less. It's a thermocline that stabilizes at that. So imagine the temperature differential just in a few centimeters is 300 or 400 degrees difference of temperature. Imagine the drastic chemical reactions that are happening in that temperature differential, okay?
Amongst what is, I mean, it's really cliche and the deep sea is definitely not a desert, but relatively islands of abundant fauna, islands of life in what is usually desert-like deep sea globe. Oh, this is so cool! What, 27 meters high? 30 meters high.
Those are towers. Okay, it's just a couple of years ago. And so the fauna that you saw there, all of these creatures mostly feeding on bacteria. Okay, and that's why you see they have all these hair structures, hair-like structures to trap whatever is floating around. But there's a whole ecosystem that develops that begins with bacteria that instead of being photosynthetic, they are chemosynthetic. All right, so it's the bacteria that feed on this ooze that comes out and transform it into organic material. They grow, and then these animals feed on those bacteria. So typically the animals that exist around these vents, these hydrothermal vents, are filter feeders. The animals are filter feeders. Okay, let's look at another one then. So there are two theories today, two prevailing theories. Each one has its own merit. That life, that soup, the original soup, the protobiont, happened either at the surface of the ocean as the ocean is moving back and forth, let's say on the shore, that would cause a cycling effect, right? The shore coming in and out, the waves coming in and out will cause a cycling effect. For the protobionts to aggregate. That's one theory. The other one is in total contrast, that it didn't happen on the shore. It happened actually at the ocean depths along these vents, where we have these conditions that are extreme conditions with the ooze coming out from the earth and encountering salt water and the drastic chemical reactions that are happening in that micro environment there. All right. What is common to the two theories is that somehow it happened in water. It happened in water, and to this day, living organisms were all full of water. All right. So let's see. We've looked at the polymerization. Yes. Yes, we saw the Urey, the thermal. Oh, okay. So this will prep us for the next, this video. It's a little longer. This video is about 50 minutes. This will prep us for the geologic ages. That will be the end. All right. And this talk's a little longer. It's about 15 minutes. A scientist talking about the transition from no life to life. And it's one of these TED Talks.
You know, this small idea can actually make an impact across the globe.
I believe there's a better way.
There's got to be a better way.
I believe we can transform the world.
There's an ad.
I can inspire others. This is something that could benefit everybody. I think this is worth sharing.
An idea that's worth sharing.
How we address gun violence in the United States. What we've really been sold in this country is fear for 40 years.
There is a model for small business success. What if these great ideas didn't have to fail? These companies didn't have to die.
This is an ad, sorry.
You will be saving 245 million pounds from the landfill. There are estimated 400,000 unsustainable across the country. But we can't wait for things that we need to get done. It's a real problem. It's real numbers. Business can do something that no other industry can do, and that's in racism. Their messages of saving communities, of inspiring change, and messages of hope, messages of peace, and also messages that make you a little uncomfortable.
After listening to the ad, he went away. It's not fair. Let me see. Huh. Look at that video. Maybe this way. Okay. Oh. I don't understand. It's here. Oh. What happened? Try again. If I leave it alone. Sorry, folks, I'm being technically challenged today. Well, maybe if we take the name. And sick. Yeah. It's a memory thing.
What I'm going to try and do in the next 15 minutes or so is tell you about an idea
Oh,
how we're going to make matter.
sorry.
come alive.
Somehow I went to Chrome.
Now, this may seem a bit ambitious, but when you look at yourself, you look at your hands.
Sorry. So this is it. Pasting row. Okay. Oh no. Let's put the ad again. Let me see how we're gonna do this. No, I don't want that. Bless you. Oh. Somebody doesn't want us to watch this. Oh, that's going to go into Chrome. No. I don't know. Here's the talk. And it gets stuck. There must be a memory issue. Yeah, it gets stuck, you see, because it's in Chrome. Okay, maybe because it's too long, it's 14 minutes, it's kind of long. So let's, let me go back for a moment to finish the lecture here on the geologic eras, just to point out what is known as mass extinctions and radiation. Mass extinction and radiations. Geologic eras are measured in millions of years and hundreds of millions of years, geologic time. So we know it's a large time scale. And they have been a sequence of mass extinctions and after mass extinction, radiation. In other words, new species flourish, come up and flourish and continue to diverse into other species until there is another mass extinction, and then after that mass extinction, another radiation and so forth. So we have these waves of speciation that has happened throughout geologic time, and that's all corroborated mostly by the fossil record. And the theory is that species radiate and radiate and radiate until there is some kind of a cataclysmic event, some kind of a massive change in the climate, which will make species, which will test the survivability of species. So until there is a drastic climatological change, species generally will continue to survive and adapt and so forth. But if there are drastic changes in the environment, then that could cause a mass extinction, especially of the less hardy species, the species that were not as flexible to survive the change, right? What that does is, with these mass extinctions, then it makes what is known as niche availability. It opens spaces for new species to evolve, because typically in the radiation phase, if you will, these species are becoming more and more aggressive. There's more competition going on, so they're fighting for food and whatnot on the consumers, right? And the producers also trying to get taller, to get more light and so forth. So on the consumer end, we're fighting for food. On the producer end, they're fighting for natural resources, basically water or sunlight or both at the same time. But there's competition across the board, right? It's natural, it's built in. Then when there is a drastic climatological change or atmospheric change or change in the crust itself, like a volcano or a mountain range or something like that, then that causes a mass extinction, which kills off many of the predators that were preying on their victims. And so that opens up new niches for radiation to happen again. You see, new species moving into those territories, following the sequence of pioneer species and then getting established and so forth. One big one was the Cambrian Explosion about 500 million years ago. 500 million years ago already we had multicellular organisms, we had producers and consumers and so forth. Most of them were aquatic. There was a mass extinction there. It's not really known what caused that mass extinction, but right after it, there was a huge radiation, which is known as the Cambrian Explosion. To date, is the largest radiation known to us, to scientists, in the Cambrian Explosion of the various radiations that have been identified. Okay, about 5 of them actually in this cycle. And but we have evidence from the fossil record, especially sediments in what is today Canada, of huge deposits of petroleum that got embedded with all kinds of fossils from that era. All right. Then the next one that was very exciting for mammals, for the possibility of larger mammals, was the extinction of the dinosaurs and dinosaur-like creatures in about 65 to 70 million years ago. Okay. And that allowed the transition from the age of the reptiles to the age of the mammals, which is now the Cenozoic, is mostly the larger animals around on land and on sea, paradoxically, are mammals. Okay. So I just have little film clips on these two. First, the Cambrian Explosion, dealing with a massive extinction and then a radiation of species there, but all aquatic. That was all aquatic. Eventually, toward the end of the Paleozoic, some animals begin to migrate into land, and we get the land animals, and plants. Plants had to go first, of course, come on land first. And then through the Mesozoic, it was mostly the radiation of reptiles and birds as a derivative of reptiles. And finally, to the point that it became what is known as runaway evolution. Runaway evolution with the dinosaurs. They just got larger and more ferocious. And then on the carnivores, on the consumers, and on the side of herbivore dinosaurs, they were just immense, and that was basically the adaptation that they had for the large carnivores, was a huge volume of mass of the herbivore dinosaurs, because the smaller herbivores just got eaten up by the carnivores. Okay. But that is a classical example of what is known as runaway evolution. For example, the peacock. The peacock basically is kind of on a dead-end route, because that tail for the male has become so huge. They can fly, but it's such a burden to carry out around that tail. See that in the wild they have a difficulty surviving, because they get taken down by just by cats because of the burden of the tail. Okay, so let's look at some clips here. This one is on the Cambrian Explosion, and the other one is on the dinosaur extinction. Let's see if we have more luck with these. Okay, looks like it.
A mere 542 million years ago, the Earth's landscape was a barren wilderness. 4 billion years after the planet's creation, no animals lived on the land. The most developed organisms were simple animals living in the ocean.
Lower volume.
Something extraordinary was happening in the seas. Over a period of 20 million years, at the beginning of a period called the Cambrian, an explosion of new animals developed below the water's surface. These later gave rise to most of the diversity of animal life that now exists on Earth. But the cause of this sudden burst of life, which geologists call the Cambrian Explosion, remains a mystery.
The Cambrian Explosion was really biology's big bang. Things went from moving very slow.
Let me pause for a moment. So these obviously are animations, but they're based on fossils that have been found on the fossil record. So it's just not making up sci-fi creatures floating around, all right? These are based on the fossils that have been found in different parts of the world, mostly embedded in tar deposits of petroleum deposit of shale rock.
Only along the seafloor without any predation. But suddenly, all the different type of ecologies that we find today, life literally exploded.
What triggered the Cambrian explosion? We don't really know. There are many different possibilities, different hypotheses, and we've got to remember that cause and effect are often difficult to disentangle.
So what happened exactly? To try to understand it, you need to look a little bit farther back in the history of the Earth, to the Ediacaran period, which preceded the Cambrian. It was during this time that the first large animals evolved, but they were shellless.
Before the Cambrian, we go into what's called the Ediacaran times. And this is an extraordinary interval where we have the seafloor populated by basically weird creatures. And quite frankly, we're not quite sure whether they're animals or something else entirely. These are organisms which seem to have mostly survived on absorbing nutrients from the seawater itself. They may not have had any gut. Some of them may be able to move. These creatures are principally soft-bodied, that is, they have no skeleton. And then they seem to more or less disappear as the Cambrian explosion kicks off.
After 45 million years of slow evolution, those weird creatures suddenly disappeared, to be replaced by those of the Cambrian. The first echinoderms appeared 540 million years ago. Starfish and sea urchins are their descendants. Brachiopods followed after some 10 million years. The trilobite, the Cambrian's most iconic fossil, joined the explosion towards its end. It showed up a few million years later. There are a number of theories about what happened that enabled these creatures to appear.
There's a theory, a hypothesis, suggesting that one of the factors that holding back the evolution of the animal is the availability of oxygen. Photosynthesizers, things like cyanobacteria, algae, plants, make oxygen and organic carbon. And once the organic carbon is buried in the sediment, the oxygen is released in the ocean and the atmosphere for animals to use.
Prior to the Cambrian explosion, oxygen levels may have been too low for large active animals to survive. But once those levels increased, larger animals were able to survive and proliferate.
Then animals burst and diversified in many, many different forms.
Evidence to support this theory of higher oxygen levels is scarce, but there may be a link to a phenomenon called Snowball Earth. This was a long period of recurrent ice ages. When the glaciers melted, huge amounts of rock were exposed and eroded, pouring nutrients into the water. That encouraged the growth of photosynthetic algae, which would have caused oxygen levels to rise. But the last of the giant ice ages ended around 90 million years before the Cambrian.
Things like oxygen levels, which are fairly clearly increasing during this interval close to the Cambrian explosion, must have been important. But oxygen was accumulating already much, much before then.
Another theory for why the Cambrian explosion occurred is that minerals became increasingly available in the oceans due to post-glacial erosion. These could have been used for the construction of shells. But Cambrian shells were made from a range of materials. This suggests that they were the result of parallel evolution in different animal lines, not a single innovation. That means something encouraged them to develop on more than one occasion, and shells are costly to make. It's unlikely mere abundance of minerals would have been enough. Could a transformative innovation in the animals themselves be the explanation? One such innovation could be bilateral symmetry, which concentrates nerves and sense organs at an animal's front and encourages the development of an efficient gut with a mouth and an anus.
We're inventing nervous systems, we're inventing eyes and other sensory systems, we're learning how to move fast, how to swim effectively. So you have a whole cascade of feedbacks, and it's really trying to pinpoint, if you like, the trigger versus all the other consequences which flowed from that initial point.
The Cambrian explosion happens after a series of events, in my opinion. First, you need to have genetic building blocks to make an animal. You also need oxygen to support animals, to support animal metabolism. But that alone is not sufficient either. You also need a mechanism to drive the system into a runaway situation. You need to make the system work very fast. And one of the mechanisms to drive the system to a runaway situation is ecological feedback.
Ecological feedback, in this sense, refers to the interaction between predator and prey. During the Cambrian, predators evolved new mechanisms, such as vision, to track down their lunch.
A good part of what's happening in the Cambrian explosion must be the motor of ecological change. And one of the really big, important gear changes, so to speak, to pursue that analogy, is in the way in which predators and prey interact. Part of the explanation for the widespread development of skeletons must be the rise of predators. And we see other evidence in terms of mouth parts, even gut content sometimes. You can see what one animal swallowed. All these things suggest that we are moving into an ecology which is radically different from anything ever before. An integral part of this is the evolution of nervous systems. And nervous systems, of course, include ultimately brains, and ultimately, of course, when animals come onto land, and sometimes even in the ocean, big brains will start thinking.
Some believe the emergence of vision during the Cambrian period caused the rate of ecological change to accelerate.
I believe that vision must have played a significant role. I think the fact that that event coincides with the start of the Cambrian explosion possibly could indicate that vision was actually the only thing that was required to trigger the Cambrian explosion. Light today is the most powerful stimulus of all. Vision is the most powerful sense. When that's unleashed on the world for the first time, it's going to create chaos.
Now, the capability to sense light has been there for a long time. Even some bacteria has a way to sense light. Many microorganisms have a way to sense light. But with very complex eyes, they can very precisely position the prey. They can view 3 dimensions and allow the predators to very effectively catch the prey. That's a very important part of the ecological feedback that allows the Cambrian explosion to happen in a very rapid rate.
Now we have this pinpoint accuracy. A predator could spot a prey, and also because it's fast swimming part and hard part itself, it could get to that animal instantly and tear it apart. So that means now there's huge selection pressures on all those soft-bodied animals to evolve defenses against this, which means really hard parts. It means shields and spines, and also hard swimming parts to swim away itself.
But not everyone agrees.
Now, there's absolutely no doubt that eyes did evolve as part of the Cambrian explosion. My reservation with it is that eyes come in all sorts of shapes and forms. And primitive eyes are really little more than what we call eye spots. They're sensitive really to sort of shades of grey and white, no more than that. And that's quite sufficient for the great majority of animals. They don't actually make an image on their eye. Whereas I think one of the dangers, if I may say so, is seeing evolution through our eyes. Is we have fantastically well-adapted eyes, which are the product of millions of years of evolution. And there's little doubt that those really sophisticated eyes only evolved a bit later.
There is one other possibility. The disappearance of the Ediacaran creatures may be the cause, not the consequence, of the Cambrian explosion. The explosion resembles the recovery from a mass extinction. Signs of a catastrophe 542 million years ago could easily have vanished by now, but that doesn't mean one didn't happen. Perhaps the bilateral ancestors of today's animals were simply hiding in the shadows of the Ediacaran world until opportunity knocked. Whatever actually did happen, though, they took that opportunity, with momentous consequences for the future.
Humans are direct descendants of the Cambrian explosion. We have primitive fish from deposits such as the Berger Shell in Canada, which we can really say are our ancestors.
From the human point of view, the true significance of the Cambrian explosion is that Homo sapiens wouldn't be here if it hadn't happened.
Okay. Still pending ours, the other one for the dinosaurs, but we're running out of time, and I'll try to find out what's going on with that other video. But basically, what we have then, let's see on the handout, the various geologic eras, right? We're looking at the transition here from the Precambrian to the Cambrian, and then that Cambrian explosion, which is a radiation of many, many species, continues. There was another one halfway through, let's say, around 250 million years ago. And then this is where now we have terrestrial plants and animals. And on land, looking at the land, is the runaway of reptiles into dinosaurs. And this one, the most prevailing theory is that meteorite that hit the Earth back about 65 to 70 million years ago. And it was hinted at over here that maybe the one that caused the mass extinction before the Cambrian explosion may have been also a meteorite. There's evidence, there's geological evidence that the Earth has been bombarded by meteorites many, many times. And it's only fairly recently when we developed an atmosphere thick enough to shield us from those meteorites. But when we look at the moon, which has more or less our age, we look how bombarded the moon is, right? And we should look just like the moon if it weren't for our atmosphere. Okay, so I'm gonna leave it there. God willing, we'll pick up next time. So let me ask, any questions or comments? No? Okay, I'll send you all this updated by email, including the link to the video, which I'm going to stop recording now. Okay? Thanks again, Father Robert. Thank you for listening.